Hello friends, this is Prashant Kadiyar. I am the faculty of the Life Science Academy, which is located in Prayagraj, and this is the premier institute for GATE and CSIR NET exam preparations. So in this video we discuss about the role of NADPH and glutathione in oxidative damage. How the cell protect from oxidative damages. Okay, so what is the mechanism behind this this protection? So first of all, you must know the what are the reactant which is responsible for this kind of reaction, oxidative damage. What are the reactant are responsible for this event, oxidative damage? So whenever the oxygen species that is produced in while mitochondrial respiration is produces its derivative, that is so much reactive in their sense. So these reactive is actually harmful for their cell. It doesn't it harms the integrity of the membrane, cellular membrane, and leaks out the cellular content into the environment. So actually some species like that is called reactive oxygen species, like superoxide, H2O2 and hydroxyl free radicals are responsible for these are the reactant which are responsible to cause oxidative stress in the cell. Okay, but how they generated, where they generated, and what are the reasons responsible for their generation? These are three main factors. They actually generated in mitochondria. First, they are generated into mitochondria. Okay, and what are the pathways responsible for generation these kind of reactant? Pentose phosphate pathway, or you can say HMP pathway or hexose mannose phosphate pathway. And what are the reasons which provokes the generation of these kind of reactant? First reason is mitochondrial respiration. During mitochondrial respiration they are produced. Second reason is ionizing radiations. Due to ionizing radiations, consumption of sulfur drugs, excessive amount of consumption of excessive sulfur drugs. They are called anti-malarial drugs also. And some toxic ingredients also responsible for generation of these kind of reactants, like DVC. Okay, now we look at come to the main mechanism, how they are generated. So when the oxygen when the molecular oxygen is O2, is undergo the reduction process. When they gain electrons, it produces superoxide. Okay, these superoxide are very reactive species, very reactive oxygen species. They further react with 2 moles of protons and electrons, which is coming when the proton is coming, when the proton gradient is established across the membrane, mitochondrial membrane, and electron is also transferred from one complex, protein complex, to another complex. There is several complex inside the inner membrane of the mitochondria. So protein 1, protein complex 1, complex 2, complex 3 and complex 4 is there. Between complex 1 and 2, these kind of oxygen radicals is generated, and from complex 3, oxygen species also, these oxygen derivatives also generated. So whenever the proton is coming coming through this pathway, these oxygen superoxide radicals are reacting with 2 moles of protons and generate H2O2. This is called hydrogen peroxide, which further undergo 2 types of reaction. It depends on the external environment also. So if they follow of normal reaction, when the cell is not harmed by this oxidative damage, so in this case H2O2 is further degraded or breaks into H2O with the help of glutathione peroxidase. This reaction is catalyzed by the glutathione peroxidase. Okay, but here one question arises: what is the role of this pathway? So whenever when we look the oxidative phase of pentose phosphate pathway, one primary reaction is there. When glucose 6-phosphate is available, it react to produce phosphogluconate in the presence of G6PD, that is glucose 6-phosphate dehydrogenase. During this reaction, NADP, that is the oxidizing agent or coenzyme, is converted into NADPH. The NADPH creates a reductive environment and activate this enzyme, glutathione peroxidase. If we look the architecture of this enzyme and see what are the actual atoms are responsible for protection from this reaction, whenever the NADP is converted into NADPH, the glutathione peroxidase have one glutathione species, GSH and GSSG. This is the reduced form of glutathione, and this is the oxidized form. When NADP is converted into NADPH, when this enzyme is available, then this reaction is proceed in this way during oxidative phase. So when this converted, the oxidized form is converted into GSH. 2 moles of GHS is generated. So in this condition, glutathione peroxidase is activated or catalase is activated, which initiate to break this hydrogen peroxide into 2 moles of water molecules. This is a protective reaction for cell. It maintains the cellular membrane integrity. Okay, so but in the reverse case, when the NADPH is not available, it converted back into NADP. Then what happens? GSH, the reduced form, is converted into oxidized form. It means thiol group that is available in GSH is converted back into sulfur atoms. These are the 2 sulfur atoms which inactivate glutathione peroxidase, and the produced peroxide and is not converted into water molecule. Ultimately, they are converted into hydroxyl free radicals. If this hydroxyl free radical is generated, then they are called ultimate killer for the cell, because when this hydroxyl free radicals interact with the biological membrane of the cell, it creates a pores inside the membrane and leaks out the nuclear component of the cell, which result into ultimate death of the cell. Okay, so what is happening during this oxidative damage? So from this reaction we conclude that the NADPH and glutathione species, that is GSH, is responsible for protecting the cell from oxidative damage. So its best example of this reaction is because of how the antimalarial drugs act on parasitic organisms, Plasmodium falciparum, which causes malaria. Falciparum. And davicine is also toxic ingredients is also act like this. When the davicine that is present in fava beans, when the Pythagoras and their colleagues went to Middle East jungle, they to formulate some kind of mathematical equations, to find out mathematical equations, they consume excessive amount of fava beans, and fava beans have this kind of davicine toxic ingredients which harm the erythrocytic cells. When the davicine, this fava beans is consumed by their colleagues in excessive amount, these toxic ingredients harm the cellular membrane, erythrocytic cells membrane integrity, harm the erythrocytic cell membrane. It is harmful for this erythrocytic cellular membrane. So in this reaction, glucose 6-phosphate dehydrogenase enzyme is not available, its deficiency is there, so glucose 6-phosphate is not converted into their derivatives. So that's why, due to the lack of this enzyme, GSH is not produced from oxidized form GSSG. That's why sulfur atom is exist, and this sulfur atom inactivate this enzyme glutathione peroxidase. That's why H2O2 that is produced from superoxide, it produces ultimate killer and harm the cellular integrity of the erythrocytic cellular membrane. So that's why several uh, colleagues uh, several colleagues uh, have found some symptoms like when the cellular membrane is destroyed, their hemoglobin is released into their blood, and it causes jaundice-like disease, which when this fava beans or devicine is exist in excessive or consumed by the colleagues in excessive amount, they ultimately causes the death of their colleagues. But they are protected from malarial disease also. When they malarial disease also, and this malarial because erythrocytic cells is already destroyed, though that's why Plasmodium falciparum parasite is not harm their those colleagues, but normal individual is harmed. So this is all about this oxidative damage. Thank you very much.